Hi, everyone. Welcome to the Scrubbing and Cleaning of Sensitive Data workshop. We are very pleased to be here. Two of the three of us anyway. We're still waiting on our third panelist, Dr. Kelly Bakulski, but she will be here, possibly experiencing the internet blip that seems to have knocked the server down, where we have the materials for this workshop at our GitLab. But once that is up and running, all of those will be available to you, including the slides and the recording from this lecture. And so thank you for MIDAS, the Michigan Institute for Data Science, for having us and for hosting what I'm sure has been an amazing symposium so far. So contained within the GitLab repository, you'll find the sample code that we'll be walking through, as well as some fake data sets that have been prepared for this workshop. If you have any questions, please post them in the chat or at the Q&A. So again, this is the Scrubbing and Cleaning Data workshop. And thank you to the Michigan Alzheimer's Disease Research Center, our employer, for allowing us to present some of what we're doing for all of you. For today's agenda, we're going to start with some introductions, go through the goals of the workshop and some of our working definitions, as well as touch on what sensitive data are and why should we care. With that in mind, we're going to take a tabular data journey through the tidyverse with some helper functions. Nick will be presenting on a non-tabular data set, and we're going to take a look at some analysis from Dr. Kelly Bakulski. So I'm Jonathan Reeder. I'm a programmer and data analyst for the Michigan Alzheimer's Disease Center. Just over a year, almost a year and a half here now. Professionally, I'm interested in developing data pipelines in order to ensure accurate and quick processing of raw data for end users, as well as project management. And my personal hobby that has been exasperated in the last work from home couple of weeks, months, has been being a cat owner. So she may make an appearance later. Nick, if you want to go ahead and introduce yourself.
Yeah, hi, I'm Nick May. I'm the data report manager at the Michigan Alzheimer's Disease Center. I'm mostly interested in the plumbing that moves data around from place to place, and particularly interested in functional programming approaches to data management and data engineering. Yeah, free time I run and I walk, and where I can I use diaresis or lots of old diacritics in the language, and I hope you get to spot some of those in my slides. Kelly's joined us, but she's a guest. I don't know if we can make her a panelist somehow. Yeah, sorry for the holdup, everybody. We've had some internet trouble.
Hi, folks. I'm happy to join you today. Yeah, we had a blackout over here in Water Hill in Ann Arbor, but I'm thrilled to be here. I'm an epidemiologist, so my primary interest is in the applications of this data scrubbing and data cleaning, which both John and Nick are going to describe to you. And one of my personal hobbies is gardening. I'm a big plant aficionado, so we're excited to be here with you today, and I think this is going to be a great workshop.
Thanks, Kelly. And I like to always start off with some thank yous. So we already said thanks to MIDAS and the ADRC, but also to the NIH. They support our funding, NIA, to NAC, the National Alzheimer's Coordinating Center, who we work very closely with as one of their 30-some centers, promoting the uniform dataset data to create a several thousand participant data set, which has been great over the past couple of years. As well as the creators, maintainers, contributors, etc., to our tidyverse, and Scala, which we use frequently in our day-to-day. And as well as to all of you, thank you for being here and choosing us to be your workshop during this time. So we have three main goals here, and the first goal of this workshop is to demonstrate the importance of an ETL, or an extract, transform, load pipeline, where data can be coming from multiple sources. Along those lines, the second goal is that we're gonna describe what protected health information is, or PHI, and highlight the ease in which that can be scrubbed in the pipeline, as well as why it's so important to do so. And then finally, to show how scrub data can be analyzed and quickly built upon for further research in terms of reproducibility, because of how important that is for moving science forward. So to start off with our first definition, we're thinking of a data pipeline kind of like this cartoon here. Amazon Web Services gave us a definition. So you are defining the data-driven workflows so that tasks can be dependent on the successful completion of a previous task. Basically meaning that you have to start from the beginning in order to end up at the end, and there has to be successful completion of each of these different parts within the pipeline. And PHI, a very important part of this workshop, or protected health information, is the term given to health data that can be created, received, stored, or transmitted by HIPAA-covered entities. And so HIPAA defines a lot of PHI. There's about 17 or 18 different aspects of what are protected health information, some of which are fairly obvious. So names, including a full or a last name and an initial. And some are less obvious. So this is a beast of a definition, but it's all geographic identifiers that are smaller than a state, except for the initial 3 digits of a zip code if, according to the current publicly available data from the census, the geographic unit formed by combining all zip codes with the same initial digits contains more than 20,000 people. Alternatively, if the initial 3 digits of a zip code for such a geographic unit contain 20,000 or fewer people, you can only distribute that information by changing the first 3 digits to a 000. So hopefully what is a more clear example than that is this little chart here. So Washtenaw County, where we are right now, for those of us here at the University in Ann Arbor, would be considered PHI. It is a geographic unit that is smaller than a state, as would the full zip code. So where I am specifically is 48105. However, because 48105 and any other zip codes starting with 481 have greater than 20,000 people in the capacity, 481 as a zip code abbreviation is not considered PHI. So when I was trying to find which of the zip codes would still be considered PHI until you change them to a 000, thankfully the Health and Human Services website maintains a list of those 17 specific districts. So for example, those starting with 036 would get changed to 000 and therefore not be considered PHI. Another PHI are dates other than the year directly related to the individual. And so those can be things like birth date, death date, or even the exam date where someone came in for a visit. So in order to scrub the full date, you can drop down to just the year. Now there's also a caveat to this. For individuals who are aged older than 89 years, you need to combine them into a singular group that basically says 90 or older. And that is because there are few enough individuals where it's more possible to be able to identify them with less information based on the year just because of their age. A lot of these are, again, more straightforward, such as phone number, fax number, an email address, a social security number, medical record numbers, or an MRN, health insurance beneficiary numbers, account numbers, such as the checking, certificate and license numbers, vehicle numbers, such as serial numbers and license plates, device identifiers and serial numbers, so the identifying number on, say, for example, your phone, the IMEI, I believe it's called, Web Uniform Resource Locators, or URLs, Internet Protocol or IP address numbers, biometric identifiers, including fingerprint, retinal print, and voice print, as well as fully face photographic images and any comparable images. And those are just the key identifiers that fall into the category of protected health information. But in addition to that, there are also other sensitive data types that you should be aware of as you're working with data for your participants. So some other examples of sensitive data include credit card numbers, student education records that fall under FERPA, as well as personally identifiable information, which generally falls more into the category of medical records, height, weight, blood pressure, things like that. And so because we want this workshop to be generalizable and be more of a resource sharing as well, this is a site that I like to go to, an example of sensitive data by classification level from Safe Computing here at Michigan, where it breaks down some of those different classified data types as high as restricted, such as credit card numbers, through high, moderate, and eventually down to low. And basically, each of these can give you an example of what is included in that and where in particular here at Michigan at least that data may be stored. So some places may have particular deals with, say, Box, where you can have a certain Box cloud account that allows, for instance, HIPAA compliant data. So those are really our working definitions, the 3 goals of our workshop today, and who we are. So now that we have an idea of what these sensitive data are, why should we care? Well, that's the million-dollar question. And as recently as the end of October this year, there was an article posted by the Health and Human Services that Aetna, the insurance company, was to pay a $1 million settlement for 3 HIPAA breaches that occurred in 2017. And you can click on the source here once we have the slides available for you to read more about it. But basically, there were 3 different instances in 2017 where Aetna had failed to take proper privacy measures. So in one case, there were letters sent to participants who were waiting for their HIV status or medication, and that was clearly visible through the envelope along with their name, first and last. So as we know, first and last name is protected health information, as is medical status based on whether or not the medical record number was there. At the very least, it was PII. So when you're working with these data, and I'm in a role where I'm working with these data pretty much every day, we want to showcase what we actually do in terms of our pipeline. And so there were a couple ways we thought about going and presenting this workshop. One was to do a more theoretical. The other was to actually cut down some of what we actually do and showcase a product and a pipeline that works. So with the hope that you would be able to take some of this information and use it as a template for your own day-to-day and for your own work. So our data always starts with a journey where we go on an adventure. For us, a data request comes in and is processed through one of our data pipelines, of which we have several. Through one data pipeline, we can then set to deliver data for analysis. And for example, through another pipeline, we might send it to an external collaborator, such as pushing data up to the National Alzheimer's Coordinating Center. So starting at the beginning here, a data request for data comes in through a link on our website, which for us, we tied to University of Michigan's Team Dynamics. Again, part of the rationale for this workshop is to showcase some tools that we use that we have learned to use over the course, at least since I've been here, and share those with you. So Team Dynamics is a ticketing system which replaced our old system of accepting data requests through REDCap. So when one goes to our website, down here at the bottom, you can go to the research, professional resources, and submit a data request form. That form takes you to a portal request for tickets, where it asks you some basic information, such as your first name, last name, what you're looking to use data for if there's a particular project, and what data you're especially interested in. And so once the investigator completes that data request form, that really kicks off the pipeline. So whether it was through REDCap or Team Dynamics, we got essentially a ping to let us know that, hey, there's a new data request that needs your attention. So for example, from our REDCap, and again, ever evolving, this person, which is fake, just requested just form A and B and selected, in our case, a random A and B form. All of these forms are NAC uniform dataset forms that we give to our participants, and these are your traditional psychosocial, 1 to 5 in most cases, Likert scale type reporting. For other ones, we have medications that we capture. We do neuropsychological examinations, but a lot of the forms are simply, tell us about yourself. So that would be some of the psychosocial data that we capture, and we're considering that to be the tabular data. If you think of tabular data, that's almost like it can be put into a spreadsheet. It captures columns and rows and sort of a nice, neat package. And like I had mentioned, we have a HIPAA-compliant instance of Box that we worked with ITS and HITS to set up for us. In the shared Box account, we do all of the work so that we can share the project folder with the data requester, kind of like a GitHub repo or GitLab when it's working. And from that, we can share with them in a transparent way. Here is the data set that you asked for. Here's the data request, as well as here is the scripts, which we generally comment out pretty well and say, here's the logic that we took to arrive from the data that we have to the data that you requested. Some other examples of data that we capture are neuroimaging and biosamples. And Nick is going to go into an ETL pipeline that he uses for this neuroimaging data. I'd also like to thank Alex from CSCAR for creating the drivers and the trips CSVs for the generalizable transportation data set. Unfortunately, our timing doesn't allow us in this workshop to go through both the biomedical examples and the transportation examples, but all of the fake data sets are available in that GitLab repo. And so once that can be accessed again, we encourage you, along with the sample code that's up there, to try and play around with some of this and see what works for you and what might work in your day-to-day. So just to outline where we're going with our data pipeline, we're going to take a quick detour and just introduce the tidyverse to those who may not necessarily be familiar with this particular quote-unquote grammar of R. We're going to extract data needed from the data request, as well as from the data storage where we keep everything. We're going to transform data from the storage in a replicable and reproducible way. And we're going to put in here the use of custom helper functions that can help us do so in a reproducible way time after time. Because it's so important, we're going to run a second check for PHI. And once the PHI has been fully scrubbed, the data set gets approved. We load the data onto a secure cloud space. And from that point, we can either further process it through different ETL pipelines, present the data, or analyze the data. So an introduction to tidyverse and what I call tidy-ish principles, which are what I follow. So the tidyverse itself is an opinionated collection of R packages for data science. And the key point here is that all packages share an underlying design philosophy, grammar, and data structures. And essentially what that means is it makes your code easy to read, easy to follow, and sort of follows the same structure depending on which packages you're using, as long as they're part of the tidyverse. It allows you to use the code in a similar way that you would for a different tidy package. And so for those of you who were able to access GitLab earlier before it went down and cloned or downloaded the Git repo to your local machine, here's where we're using the data request template.r script in order to obtain important information from the data set. And so the data that we're going to use for here is this data request example.csv, also found in the repo. So as a reminder, here's the data that requested, just a simple form A and B, and the sample data has a form A, B, and C. So you start with installing packages like you would for other R libraries, but you can do them all at once by just installing tidyverse. Load the library. And here we have two sources. So source basically calls in R a different script at a particular path on your computer and basically just kind of runs it in your current environment. And what that does is, A, it allows your current script to maintain a little bit of cleanliness. And it also allows you to grab different objects that you don't necessarily want within your own script. So for example, when we access REDCap, we do so through the API. But for security purposes, we store that API token, at least our private token, in a configuration file. And so I'm going to ask Nick to just kind of pop in here and just explain a little bit more concisely than I could what that means.
Yeah, so if I wanted to pick up John's code and run the same process that he does to extract some data, clean it up, and then like send it off to some file share service. What makes that possible without sharing our credentials publicly online is using configuration files that we keep stored like locally or in some other file share system that only we have access to. The configs file is loaded by the script, reads in our personal unique string tokens or JSON web tokens or whatever they are, and then is able to run the code using our personal credentials to access the services or the data that we're requesting in the script. So does that capture it mostly? Okay, cool.
Thanks. So again, all of this is available in the GitLab that will hopefully be back up shortly. I tried to comment out pretty clearly and in more detail than I would for myself for most scripts what we're doing and how to sort of read it. So for this example, what we're going to do is we're going to use one of the reader library functions, read CSV, that's part of the tidyverse. And we're going to read in the CSV and save it as an object, df_dr, or that's shorthand for data frame data request. We're going to basically make a copy of df_dr in df criteria, but to df_dr, before we copy it over, we're going to do some translational things. So this gets more into sort of transmuting, but what we're going to do is basically from this CSV, we see that the dates in particular are in kind of a weird format that doesn't really represent a date consistently. So we need to make the date variable as a date, and there's the tidy-ish because as date as a base R function. We're going to take from date, which we know is in a format, if we're able to look at it, short month, short day, short year. We would then arrange it and take the top or the most recent from date. And so when we start with dfdr being read in, and it looks something like this, where the underscore 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 is REDCap's way of creating a checkbox variable. So 1, 2, and 3 are all checkbox options for data request 14, and a 1 indicates checked, whereas a 0 indicates not checked. After we run through our code down there from dfdr to make df criteria, we end up with a reformatted date and just that most recent request. And basically that allows us to say, okay, here's the most recent one we need to get started on. So we're going to take that information and we're going to store some of it in a separate variable that we primarily use when making working directories. And so basically what that means is we want to create a folder on our box instance that corresponds to the data request that we're working on. So string C, or str underscore C here, is a function from the stringer library, which is again part of the tidyverse. And from that, we're going to get the date, the last name, and the first name from our df criteria data frame, separating each of those elements by an underscore and storing it in requester. And so if we were to look at requester, we can see that from this, we now have date, last name, first name. And that's sort of a convention that we just tend to use. So next part is to extract from the data request itself. And so from df criteria, we're going to make df forms requested. And what we're going to do to df criteria in the interim before we can get df forms requested is we're going to mutate. So we're going to make a new variable called retain form A, B, and C. And each of those is going to be basically an if else, or in this case, a case when. So this is just something I like to do personally, is put some of the numeric indicators into words. Depending on who you're delivering data to, this may or may not be appropriate because you have to keep PHI in mind. And when sending data to an external collaborator, it should be coded as much as possible and sent along with a code book. So basically here, if drq14 underscore 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 1 has a 1, we're just going to call it get form A. Otherwise, which is indicated by this true, we're going to say don't keep form A. We're going to repeat that in this case for A, B, and C. We're going to get rid of some of the excess variables with this select. So we're going to say deselect drq14, 1, 2, and 3. And then we're going to use reader again in order to write the CSV along with stringer to create the path where we're going to write it to. And so if we look at df forms requested after this transformation before it writes, our data frame looks like this now, where it's date, first, last, get form A, get form B, and don't keep C. And there we go. We've extracted data from the request. The corresponding extraction part that we do is getting data from our REDCap instance, and that's basically just pulling down all of the REDCap data. Then we check to see what has been extracted, and then we transform the data. And we want to make sure to do so in a reproducible way so that it can be consistent. Basically meaning when I run this code, on the same data, I get the same results. If I run this code on data that's changed slightly because we've added rows or we've added columns, it still occurs in the same way, but now we have those added rows and those added columns. And this helps with reproducibility. So an example of a transformation can be found in the R scripts data transformation on the GitLab, and the corresponding data download example gives some fake data of what we might get from our REDCap instance. So for example here, just to quickly walk through this huge thing of code, we're selecting from a red CSV certain variables. We're going to take the subject ID, the exam date, the birthday, month, year that have all been stored in separate variables, gender, ethnicity, race. We're going to use filter, which I believe is dplyr, to say, take any exam date that isn't missing. This happens sometimes based on the structure of your data, where there might be a row for the subject, but they haven't actually completed a visit then, and so you don't want to include that. That's going to be more important when you're transforming in terms of pivoting, either using pivot longer or pivot wider to make a longer or a wider data set, which are used for different types of analyses. Similarly to before, we want to take the funky exam date and we want to reshape it by saying that this is the format, but we want it to be a date, and we still want to call that exam date. So we just want to overwrite the old exam date with the new. We want also a birth date here, and we're going to do that by concatenating or squishing together the year from birth year, a hyphen, the month from birth month, a hyphen, and the day from birthday. And what that's going to do is it's actually going to give us a format that can be readable as a date and change the birth date variable, or create in this sense, because we didn't have birth date to begin with, into an actual date type. So that's important because, remember, dates are considered to be protected health information, and so we don't want to be sending exact dates. Now, we can send years in the most case, except for individuals aged over 89, which does happen quite often in our data sets because of the population that we're working with, at least. So what we tend to do is just create an exact age off the bat and don't have to worry about maintaining those dates. So we're doing so here by finding the difference between the exam date and the birth date in number of days and dividing that by 365.25 to get age in years. Some other examples of what you can do are you can recode. So we had gender being 1 and 2. We might want to recode that to be male and female. We might want to rename some variables. So gender, we might want to rename to participant gender. Again, depending on who this data is going to be presented for, it may make sense to have a little bit more English readable. And finally, we want to drop some of these. So we don't want the exam date, we don't want the day, and we don't want the month. And so going through all of that from the fake data yields something that looks like this. So we've got a data frame now of IDs, birth years, the relabeled participant gender, the recoded values for gender, the ethnicity, the race, birth date, age, and the exam year. And so your data's been transformed. And don't worry, we're going to return to that birth date. And so doing that time and time again can be challenging to get it exactly the same in the same way, and also be time consuming to write that much code each time, every time, for doing what in our case is a lot of repeat tasks. So helper functions, which you've been using all along through the tidyverse, are functions you design that you can source to complete a task in the same way. And so custom helper functions are those that you write yourself. This makes future work quicker and also helps with reproducibility. So in the GitLab, you can see the helpers.r script. basically has all of that code that we did before for a transmute or a transform. And then we're just going to store it in a function called readFormA. And it's going to take a CSV, where in this case, the default is a U3 object, which is just what we called the CSV that gets downloaded from REDCap's API. And it's going to output DFA1. And so basically, every time from this point on that we need to rerun all of the same transformation, we just run this custom function, readFormA. So we've transformed, we've extracted, and we're getting ready to load the data into our secure cloud. But as you may remember, we had birth dates still in there, and it's important to remove PHI before delivering your data. So there's the offender. And helper functions, custom helper functions, are also really great here. So we posted a function that we use actually in our day-to-day work called scrub PHI. So this function gets passed a data frame, and you can specify what the PHI fields are based on whatever data set you're using. We have a pretty extensive list for the NAC data, but depending on if you're using a data set with other PHI, you would want to include those. You would want to include the exact field names. It creates an object from the column names of the data frame that you passed, and then basically says, take a look at those names. Now look at the names in PHI fields. Are any of those columns the same? If they are, get rid of them with dplyr and put the output into a scrubbed data frame. So I don't know about all of your work, but in ours, any data frame called scrub data frame is pretty out of the ordinary from anything else that we would be naming things. So it's a pretty good standout for when we know that the data has been scrubbed. So we've transformed after the extraction. We've double checked for our PHI, which we know to be those 17, 18 factors, plus any other sensitive data that you may be working with. And we push it onto our secure cloud space with, again, reader. So we write the scrub data frame as a CSV to whatever the destination is, the folder that we created from earlier on in the data request extraction. And as I had mentioned before, we have a secure instance of Box that we had worked with ITS and HITS to set up for HIPAA compliant data. So our data is loaded, and that is essentially an example of a real-world ETL data pipeline. So again, we extract, transform, write some custom functions to help us with reproducibility, double check for PHI, push it up to the cloud, and then it's ready for further processing, presenting, or analyzing of data. And so with that, let's take a quick break and come back in a few minutes. And Nick and Kelly will take over. Nick will talk about a new ETL pipeline that he's been working on for non-tabular data. And Kelly will show us some cool analyses and how to work with the data team.
Just in case you didn't, John, I set a timer for 5 minutes.
Yeah, that was really great, John. I loved how you walked through the code with those practical examples. You're gonna turn me into a tidyverse coder after all.
Tidy-ish.
Yeah.
I think like our studio's documentation is like almost reason enough to use it because everything's so pretty organized. Yeah. Thanks so much. There's about 20 seconds left. I guess we'll wait till the time's actually up in case people need it. Okay. Hi, welcome back, everybody. We're going to get started. Yeah, if you, John, if you could click to the next slide there. Yeah, welcome back, everybody. Next again. So in this next section here, I'm going to talk about non-tabular data. And as John mentioned earlier, tabular data, we can take to mean anything that looks like it's a spreadsheet. You know, you've got one record per row and a number of columns that are fields that represent, one data point. If you could click next. Of course, not all data that you may touch in your careers is going to be nice, flat, clean, tabular data. Some of it's hierarchical, like JSON or XML, and may contain PHI, PII, or any other kind of sensitive information. Click next. So here in this JSON file, I've highlighted in red some data that may be some pretty intuitive, right, like name and age, but then also the dates that these measurements were taken, these impedance measurements were taken in a data set that we've had to do some work on. There are, it's kind of the equivalent of an exam date. And so something like that would have to be scrubbed. Next slide, please. Similar in some ways is the metadata that's in DICOM files. DICOMs are a standard for transferring medical imaging data between machinery, basically. Along with the data itself are the images, but the metadata or the header data has a lot of text that may also contain,
So it looks like our internet troubles are back for both Nick and Kelly. Seems like they had both dropped out. So I'm gonna go ahead and just try to walk through what some of Nick has been working on primarily until he gets back, hopefully soon. So as he had mentioned, the DICOM file is basically a standardized file for transmitting information between machinery. And so the metadata contained within that file may contain PHI or PII. So in this instance, the name, ID, birthday, sex, age, and weight. And so similar to what we talked about earlier, another example of an ETL pipeline would be to obtain the imaging from the MRI, to, in this case, extract or create a copy of the data. When we transform it, we're de-identifying here, similar to what we did before, and also zipping it because of the many files that are produced from one MRI session, and loading it or uploading it into NAC's Amazon S3 bucket, which is how they have decided to store the MRI data. So you can see here that these are two file trees. One is the source that contains 3 patients, patient 1, 2, 3. Some have repeat scans, and within each of those scans are several DICOM files. Our target, where we want to put the data to, in this case has patient 3, but is missing patients 1 and patients 2. So basically, we want to copy patient 1 and patient 2 data over. Kelly's back. Copy patient 1 and patient 2 data over into that same directory so that it contains all 3 patients. So we, again, are copying patient 1 and patient 2, but before we push that over, we need to de-identify them. And so we have a config file where some of the variables are specific to NAC, and so we've preserved those for NAC. Other of the variables are contained here, and some of those, this is a continuing list, some of these we need to de-identify. So we need to find the name, birth date, sex, etc. So in this instance of the copied DICOM data, it contains PHI. So the patient's name, birth date, sex, age, weight, etc. are all still in there. And the goal is to essentially blank these out. So you can see here in the bottom right, patient's name becomes an empty string. Same with birth date, sex, age, and weight gets converted to none type. And this is the DICOM file that we would then push up into NAC because it does not contain any of the PHI or PII from in the metadata. And then the final part of that is to zip it so that we can take all of the many DICOM files within each of the many scans for each participant and just plunk it up as one nice zipped folder. And then from those zip folders, we push them into the S3 bucket, which requires a different sort of file structure. So as you can see here, this is more like a directory tree, whereas those are just kind of files. So the problem that we're trying to solve is we want to read, update, and then write tens of thousands of DICOM files in a deep file system tree, so a very deep structure. And we want to include only certain names. Here's Nick, thank goodness, because I do not know what I'm talking about with this.
Hi guys, I'm terribly sorry.
Hi, I don't know how far back you want me to go. I attempted to get through the different file structures and what you were doing and got here.
Yeah, thank you so much. I really appreciate it. My power flickered again and I lost the connection. So, yeah, did you go through the problems already?
Just started to.
Okay. So yeah, we want to read and write tens of thousands of DICOM files that are deeply nested. We only want to include certain directory names, certain directories with a maximum number of DICOM files, and certain DICOM file names. Also, we need to be able to look into the DICOM files themselves to get specific metadata about the images. So with the first 3 criteria there, the directory names and size and file names, we could use a tool like Unix's rsync utility to sync between a source and a target directory tree. But since we have to reach into the DICOM files themselves and read metadata and filter against that metadata, something like rsync wasn't an option. If you could click next. So some of the solutions to these problems were that we needed to find a performant language with a large ecosystem, including libraries that allowed us to read and edit, write DICOM metadata, and then also we wanted to be able to use load our de-identified data, DICOM data, to the S3 bucket. Some other criteria. So we also, because we only want to copy certain files, I wanted something that made it easy to create a custom data tree structure, and then also to write simple methods that could recurse through the tree. And where I landed was using Scala because it's object-oriented, much like Java. It's functional and it's concise. So writing any custom recursive methods on the tree data structure that we're using throughout the solution is pretty easy. And then also because it is a Java virtual machine language, meaning it compiles to Java bytecode, we can use any library or package that's in the Java ecosystem, so which is well established, very old, and huge. So there's actually more than one DICOM library option, and of course Amazon makes publicly available its software development kit for writing things to the cloud. So just as a quick glimpse, underneath this, the text there, there's sort of a simplified file system tree with directories as square nodes and files as circle nodes. We're going to use this to illustrate some of the ideas of the implementation of working our way through this tree, and eventually doing the extraction, transformation, and loading work for this non-tabular data. So next slide, please. So where we need to get from is we have the source tree, which John walked you through, and this is just a different sort of illustration of the same data. And then we have the target tree on the right in cell 2. And you can see that the source tree has more information. It's got data that, in the red nodes and then also the green nodes that need to be eventually copied, de-identified, and zip. So how do we get from 1 to 2? If you could hit the next slide. And then again. So we start with, in that little line of code, we see that to the right of the equal sign, the assignment operator, we start with the source tree, which is now in both cells 1 and 3. What we will eventually arrive at will be assigned to the variable tree to copy DID zip. You could click next, please. And then in this filter that we apply, so we apply a filter to all of the child nodes in the the gray directory node that we have a handle of. And for each node in the tree, we just check to see whether or not it is in the target tree. And if it's not in the target tree, so if the target tree does not contain the node that we're currently working on, then it gets kept. If it is in the target tree, then we get rid of it. So keep your eye on the tree in cell 3. You'll see that the target tree nodes disappear when John clicks next. So those go away, and what we have remaining is the difference between our source and target tree. If you could go to the next slide. So now we have this, the tree that represents the file system that we want to copy, de-identify, and zip. So that's our extract and 2 transform steps for this non-tabular data. If you could click next. I'm going to dive down to some implementation details, but what this illustrates mostly, if you don't, if you know, the object-oriented approach to solving this problem doesn't make sense, what this illustrates is that this approach is modular and applies this ETL framework. So in the first step, we have this tree that we need, that represents the file system that we need to copy from one place to another. All of the details for how we need to copy, the files that are represented here to a new place are wrapped up in an action class. Here specifically a copier class, right? What this class does is it implements the Java file visitor interface, which is a pattern for interacting with directories and files on your the machine that you're working on, be that your local laptop or server or whatever. So that class captures all of the information in one object, and then that object, the copier object defined there, is passed into the method that only has side effects in that it operates on the tree that you see represented. It copies the files and directories that are in that tree into the location that's defined in the copier object. Okay, so what we've done is we've taken one step of the process and packaged it into an object, and then sent that off to do some work on the tree that we need to do work on. So John, if you click next, now the next step is to de-identify. So now we're scrubbing all the information out. We have the same kind of action class, and we use the same pattern to do a similar kind of job, but instead of copying files from one place to another, we're now reaching into the DICOM files that are at the bottom, at the leaves of this file system tree, and then scrubbing out any PHI or PII that doesn't need to be a part of that. And if you click next, the same thing goes with the zipping. So now we have this tree at the, say the first level where you see the green square dir node and the red square node. We want to take that directory and zip up all its contents into one file. And all of the particulars of that, like what level in the tree we're zipping at, whether or not we're doing tarballs or just zips or whatever, all of that is packaged in the action class that is zipper. So if you go to the next, we've now copied, de-identified, and zipped, but now our file system tree has changed slightly because instead of having directories that go all the way down to files, right under our root directory, we now just have zip files. And so we need to update the tree that we're working with, and then we'll apply the same action class pattern to this tree. And John, if you could click next. You can see the code largely looks the same. A lot of the implementation details are hidden, but this is almost exactly what it looks like in the code itself that does this work. So when we go to upload these zip files to the S3 bucket, this pattern comes in handy again. So if you could click next. So yeah, through this process, we've taken non-tabular data. It's a little bit more difficult to access and work with, but the point here is that we used the ETL pipeline framework to solve, the same problem of scrubbing sensitive data out of or sensitive information out of data that we have to do something with. And then also, much like John explaining that, his use of helper functions to do something over and over again, but in small packages, what I've tried to illustrate here is how, a modular approach makes something, reproducible when you do it. So if you give it the same data, right, you give it the same input, you get the same output every time. It's more extensible. So if I wanted to add another step in this pipeline, I could use the same pattern that I used for the copier, de-identifier, zipper, and uploader. And then it's also, because they're in smaller chunks, they're much easier to understand. and then also write tests against so that you can ensure that what you're doing is exactly as you expect. If you'd like to check that out, there's a public mirror of the repo at that URL, which again will be included in the materials once the GitLab page is available.
Thanks, Nick. That was really fantastic. There's so many interesting data structures now that we have access to, whether genetics or these neuroimaging you were describing, where data are not as simple as a spreadsheet format now. So thank you for walking us through that. I really appreciate it. I have the opportunity in this next phase of our discussion to represent kind of an end user experience and talk about some best practices with data analysis. So broadly, recognizing that my lens is coming through a clinical perspective. When we're thinking about research questions, so what do you want to do with the data? I broadly have 3 buckets or bins here. So the first, we might be looking for upstream factors. What are risk factors associated with a disease? Those might be environmental, they might be social, they might be genetic factors. Then in click to 2, we see that the next bucket of research questions might be looking at outcomes of the disease. So what types of interventions or treatment could we do? Are there caregiver responses that we could encourage? So those are looking at downstream factors. And then the third major research question bucket, if you click there, is talking about methods development. So how do we improve our research by improving the metrics we use in our research? So these are kind of the 3 main research questions that I'll touch on here. And you could think about how this could apply outside of health as well, in terms of upstream factors, downstream factors, and methods questions. And one of the things that I've come to appreciate over time is that really asking an effective research question saves a lot of heartache in the long run. And making sure that your research question is narrowed and measurable makes your interactions with the data managers and the data core so much more effective and productive for everyone. And there's all kinds of criteria you can use to evaluate a research question in terms of these metrics. One set are these finer criteria. If you click, that includes a checklist of making sure your research question is feasible. So do we have the sample size in this data set? Do I have the technical expertise to analyze this data? Do we have the ability to manage this data? Also, is this question interesting to me? Am I going to stick with it all the way through? Is it novel or something that needs to be reproducing as a previous study? Is it ethical? This is certainly essential in the clinical health research. And is it relevant to the disease at hand? So next, here's an example research question I'll walk you through one we just did recently. We were curious, are medical conditions in early life prior to age 16, are they associated with your odds of dementia in late life? And I'll walk you through my interaction with the data core through this. So my first step would be to make a data request. And John described this from the perspective of the database manager. And here, as an end user, I've come to the website here, the Michigan Alzheimer's Disease Research Center data request website, but your database of choice may have other interface. In general, most of these websites are going to ask you for your IRB status. That's the Institutional Review Board, which is responsible for making sure your question is ethical. They'll often ask you for the number of your approval to make sure that you've considered that and have maintained ethical standards. They'll often ask you what your data structure desired would be. Do you want a wide data set or a long data set? Do you prefer your data object to come out in an Excel format, etc.? They want it to be in a format that you're ready to analyze it in. They'll often ask you also about your inclusion and exclusion criteria. So do you have age restrictions? Do you have restriction on prior conditions? Are there certain factors that's going to affect who they should put in your final data set? And then they'll also ask you about variables of interest. So John was mentioning certain forms. And if you click, there's often at this level a high degree of communication between the researcher and the data core, because they'll know what forms the certain variables are in. They'll have variable names to recommend that you request. And really having this strong communication between the researchers and the data core or the data manager is essential to ensure that though John and Nick have a very reproducible pipeline, that they don't have to go through this endlessly, so that we can capture the right variables at the start of the process. So if you click. Here's an example for that research question of early life exposures. So the data core would have pulled the survey data at the first study visit. This is a longitudinal example. And if you click, they would have also collected the cognitive testing data at the last visit. And then if you click, they would have scrubbed the PHI from that, merged those two visit data together, and then delivered me the data product so that I could begin analysis. Click. So here, based on the research question, we evaluated that multivariable regression would be appropriate to answer this question. Click. Just like it's important for the data managers to have reproducible workflows in terms of making sure that their results are able to be assessed by other colleagues and they're able to regenerate those findings. It's also equally important for us on the research side. So we have all of our code available on GitHub and we make sure that for publication, we make that publicly available for transparency of our results. So all of that work came together in terms of designing the research question, requesting the data, producing a reproducible statistical pipeline. And here's the culminating here with this plot, just showing that yes, indeed, we were successful in achieving that analysis and identifying that some of these early life conditions on the y-axis were associated with dementia. And so if you click, here's another example research question that's going to include some of this more complex data that Nick was talking about. So here we're curious, are cholesterol levels causally related to cognitive impairment? Click. Here it was a cross-sectional data preparation, so pulling the cholesterol biomarker levels that's measured in blood into a table. Click. And then we have the cognitive testing data, as we saw before. And then if you click one more time, here's a more complex data type. Here were genetic data, which originally came in a non-tabular form and had to undergo a high degree of processing in order to be able to be combined here. And then the last step, click, is to merge these data together, scrub out the PHI, and deliver this to me as the researcher. Click. And again, we evaluated the right statistical model for this research question. We were going to do multivariable regression, and then to do that causal inference testing, we added a Mendelian randomization piece, kept all of our code available on GitHub. If you click, because we were using genetic data, we were not permitted to have that data on our personal computers due to ethical standards, as well as these are large file sizes. So all these analyses had to take place in a secure cluster that's approved for de-identified genetic data analysis. We were working with the University of Michigan Great Lakes Computing Cluster. We were able to complete this causal Mendelian randomization analysis using this workflow. Thanks. Next. And then this last example, I'll just show you one more methods development example. So those of you who are interested in research on research, so how do we improve our research? We can also query these databases, and if you click, there we go. So here's a methods development question where how do we best summarize cumulative genetic risk for Alzheimer's disease? Our genomes are vast. They have over 6 billion positions in them, and we can have inter-individual variability at very many of them. And many of those positions are also associated with Alzheimer's disease. So how do we best optimize or summarize or aggregate across the whole genome someone's cumulative genetic risk for Alzheimer's without just considering one point at a time? How do we put all of that together? So if you click, here we put in a data request and they process the participant genetic data table. Click. They also process the participant neuropsychiatric table. So we need to know, does that person have Alzheimer's disease? And then one more click. And then we have a summary table of someone's Alzheimer's disease genetic associations. So this is an external data source. This is not collected on our participants. This is from a publication. So in another independent sample, how associated are different positions with Alzheimer's disease? So this is our source of our weights. And then one more click. This is the important PHI scrubbing, the merging, and the delivery step, which happens. So anytime we're doing a research question, generally, there's going to be a combination of data types, and the data core is really working integrally to create a composite data set that makes these research questions possible. So click. For this analysis, we are taking all the positions where someone has a genetic variant. So here's just one representative position. And then here is acknowledging that people have genetic variability at many, many positions. That's kind of like my bucket of genetic variant positions. And then we're weighting them, click, at all of these previously published associations with Alzheimer's disease. And if you click, this results when you create a weighted sum of everyone's variants, you get a single cumulative genetic risk score per person. Last click. And when you look across a population, it's generally normally distributed. So we end up with a value per person that has some range throughout the population, where some people have high genetic risk for the trait and some people have low. If you click. With this methods development, we acknowledge that all of these genetic positions, everyone's going to make different choices in this method. So people may have different preferences for how many genomic positions to include, what threshold of association should be considered with the outcome to include. So we wanted to make this more flexible, not just provide, here is the one true score, but here's an opportunity for other people to change the parameters on those scores and see what happens. So if you click, for this process, we generated a Shiny app, which is publicly available on the website, to where folks can change all these inclusion parameters and see how the scores fluctuate over time. So this is an interactive way of bringing your methods development research and making it flexible for end users. If you click. And acknowledging that over time our research questions grow. We're drawn to research because we're inherently curious people. And generally when we answer one question, we're curious about 5 others. So it's very common to request amendments to your data sets or a second data set that includes new variables, new participants, new study visits. Click. And those further data requests are really facilitated by this hard work through groundwork that John and Nick have described with this reproducible workflow. So when I come back and I ask for, hey, I don't just want cholesterol levels this time, I want hemoglobin A1C levels. They can reproducibly add additional variables to my data set in a way that it makes the format consistent when I receive it. So it's easy for me to feed it into my analytic pipeline. And just encouraging that really strong communication between analysts and data managers is key to making this happen. So click. So my key points on the data analysis side is that we need to make sure our initial starting research questions are narrowed and measurable. That helps have a focused initial data request. And having strong communication between the data and the analytic team helps facilitate those success of those initial requests. The data team have standards for reproducible workflows to prepare the data. And then on the analysis side, it's up to us too to also have reproducible analytic pipelines. And also for the data team to expect these follow-up requests so that we can do follow-up analyses and really grow these partnerships and continue to ask great research questions. Thanks.
Thank you, Kelly and Nick, for your presentation before. Those were awesome and really highlight the relationship that we need to have between analysts and data cores, and sort of at least give, you know, the other side of things a glimpse into all of the groundwork that really goes into doing some of these things so that analysis can happen and science can move forward. So just to recap on our goals, they were to demonstrate the importance of an ETL pipeline for multiple sources. And so we've shown that with a tabular and a non-tabular data set. To show how easy it is to really remove PHI. And so again, PHI is essential for a lot of centers and a lot of researchers in order to be able to accurately capture information that belongs to a particular participant, and as well as to follow those participants over time for longitudinal studies. But it's data that they are giving us that we need to respect and that we need to keep private, both legally and morally. And so with a reproducible pipeline, it's pretty straightforward in terms of sending it off for analysis. And like Kelly was saying, building that relationship with the analyst so that when a new question comes up, it's easy to say, okay, here's what you had last time. We can give that back to you and add these new things in a way that you're able to just start working on it without a lot of back and forth. So we talked about PHI to find those 17, 18 different types, talked about some other sensitive data, went through methods for scrubbing PHI from both tabular and non-tabular data sets, and showed a reproducible workflow to share data for applied data analysis. And again, we apologize that GitLab is down. We are arguing with the internet today and it's not working out in our favor. But once that comes back up, we hope that you'll check that out, some of the sample code, some of the sample data, and please keep in touch. So we'll take the remaining few minutes to go through any of the Q&A that Nick has been graciously keeping up for me because I can't see anything on this one screen share. But thank you to all of our partners and to you for attending.
Yeah, to echo John's thanks. Thank you so much, everybody. I've still got the Q&A up if other folks have questions.
Nice. Nick, would you mind just reiterating a few of the prior questions that came through? Because maybe some of those, I know folks were eagerly asking for advice what to do when when the University of Michigan ends its contract with Box for HIPAA, and I think our group is planning to move largely towards Dropbox in the future.
Right. Yeah, the use case depends on, every use case will be different. We're largely going to be using Dropbox. I don't have access to the link I shared earlier because when I lost my connection and then it came back up, I lost the Q&A section. But yeah, I'll look through to see if I can find that web page again to share with folks.
People are also curious about those Scala functions you were talking about, Nick, about how efficient are copier and de-identifier, these type of workflows. I think people are interested in this new software.
Yeah. So the question about the efficiency of the copier and de-identifier classes, de-identifier is pretty quick because it just crawls down through a tree. When it finds a file or when it finds a directory, it skips it. When it finds a file, it goes in and then either empty strings or nulls or zeros out any element or attribute or field, whatever you want to call it, that we've identified in our config file should be scrubbed in some way. That's pretty straightforward. For the folks who are interested in talking about I don't know, data structures and algorithms on those, I would love to pick your brain. Right now, as I mentioned in the Q&A, the copier copying something isn't terribly efficient because in order to do basically the filtering that I demonstrated in that slide earlier, it has to check the target tree over and over again. I'm thinking about checking using what are called tries or what are they, prefix trees to speed that process up a little bit, because right now, worst case scenario is that it could have a big O n squared runtime in order to do that copying. That can be overcome, be overcome in a hacky way by using the config regular expressions that pick out the certain directories and stuff like that to break the jobs down into smaller bits. Because obviously big O n squared can be a pretty bad runtime. Yeah, please reach out. I put my email in the Q&A, and if you have any questions about that, I'd love to talk more about how I've implemented things.
Yeah, John, do you have access to any of the other Q&As from earlier in the session?
It doesn't seem to be popping up for me.
Okay.
Shiba Muli, thanks. I'll give that a shot. Tree sampling. Thanks for the suggestion.
Well, thank you all so much for your attention. John, do you want to send us out?
Yeah, I don't see anything else in the chat or the Q&A, so thank you so much for your attention, and you know, even though this is a pipeline that we've been using and works for us, please keep in touch, and we are very open to sharing, you know, a little bit more of what we do and absolutely listening to what some of you all do. This is far from perfect, and open source and sharing is really the way to go. So thank you so much and enjoy the social hour.